welcome to another episode of the SNEV podcast. My name is Liam Hourigan, and today we have on as a guest An Hendrix. She is a bacterial EV researcher, and she is the original creator and project lead of the EV Track project. Now, this episode, I would have to say, is probably my favorite episode of the podcast thus far. That's because it has this focus on the EV Track project, which is a meta-science project. It sort of looks at how we conduct science, the processes, the methods, from this top-down meta perspective. And that is, sort of my personal interest. And I bring this up now just because I also bring up a publication that I, collaborated on with An, and in that we used a bunch of EV Track data. So, it kind of has some bearing on what we're talking about here. So I didn't, just bring that up for shameless self-promotion. But now that we've got all that out of the way, let's hear from An Hendrix.
Well, thank you, Liam, for this very kind invitation. It's really a pleasure to contribute to this SNEV podcast session. My name is An Hendrix. I'm full professor at Ghent University in Belgium. And my research team is currently focusing on different topics, which really range from standardization and reproducibility efforts towards studying the biogenesis of these EVs, and then ultimately also developing them for biomarker and therapeutic applications. So it's a really broad range that we are covering. But I think, most efforts during the past couple of years really related to standardization and reproducibility tools, which we have been very actively involved in developing these for the field.
It's very important work, and that's exactly why we've got you here. First, you maybe want to just tell us a little bit about how you got into EV research, because that's always fun to hear about all the different pathways people took.
Yeah, so then we go back to my PhD. My PhD actually kicked off with, it was focusing on cancer research, and I was basically mainly interested in studying the communication mechanisms used by cancer cells. And back then in our lab, there was it was co-hosted with the dermatology department, and they were studying Rep27 GTPases related to different dermatological diseases. And we came up with the idea, why not explore the role of Rep27 GTPases in cancer cells? So during my PhD, I worked on different Rep GTPases, trying to explore what their role is in the contribution to different steps of cancer progression. And there we basically identified that the Rep27B GTPase stimulates the growth and invasion of estrogen receptor positive breast cancer cells. And we also made a very nice clinical association on patient materials, showing that there is a link between the abundant expression of this protein on primary breast tumor tissues and the development of lymph node metastasis and poor prognosis. So basically, we published this work in JNCI in 2010, and exactly in the same month, there was a publication by Matthias Ostrowski and Clotilde Thierry's group showing the role of Rep27 GTPases in the release of exosomes through the transport and fusion of multivesicular endosomes with the plasma membrane. So then, of course, we immediately made the link, we should look towards how the Rep GTPases on these patient materials are involved in the release of exosomes. And then, of course, I started my postdoc and recruited my first PhD student. And we immediately thought, we want to study exosomes, they were called back then. But how are we going to isolate them? Because there was not that much knowledge. You basically had a differential ultracentrifugation and exoquick density gradient. And we were not really sure how to approach, which method, and would it impact. And this is actually a decision, if I look back to that really defined my further research career, because although we were addressing a really biological question on, we observed the higher expression of the RAP27 GTPase in these cancer cells, but also in patient materials. And we were really wanted to explore how these EVs contribute to this process and mechanism of cancer progression. But by just asking this simple question at the beginning of my postdoc career, it completely turned towards standardization, reproducibility, how to isolate EVs from the different biofluids we were studying. So this is basically how it all initiated.
Yep, people are still asking that question now.
Yes, indeed. It's still a very huge challenge. And if you even, many people still come to me and they ask, but I'm working on this biofluid, like which method, which gold standard do you recommend? And that's still the overall idea is there is no gold standard. It really depends on, first of all, which biofluid are you working on, but also what are you aiming for? Like, what do you want to show? What's the goal of your study? And this all really defines, how you have to set up a protocol. So it's a little bit more complicated than everyone starting to work on EVs originally anticipated. But at least there has become good guidance by means of the ISEV guidelines, but also different tools that like the EV-Track tool that we have been working on, that really help to synthesize and to inform and make researchers aware that at least when you select a method, that it can really have an impact on the downstream analysis, and that you should properly consider this. And I think at least the fact that there is an awareness is already a very big step forward compared to 15 years ago.
Yeah, I think, you're speaking to a student network, and maybe that's something we take for granted now. I don't know what it was like to start back then when we didn't have MySev to look at, because that's what you looked at when you first start in the field to get a feel for everything. So you run a core facility in the University of Ghent now, an EV core facility. What sort of methods do you use? And, maybe do you want to talk a little bit about
so the core facility is very recent. So we basically moved to a new research building, but the concept of the research building is that all different areas have to be clustered in core activities. And of course, one of them, when I was moving into the building, was the core facility on extracellular vesicles. So we have multiple teams at Ghent University that contributed equipment to build up this core facility. And what we really try to provide is, first of all, really enable and stimulate access to technologies so that you can at least perform extracellular vesicle research in compliance with MySev guidelines. So any basic technology that you would need to do particle analysis, making use of nanoparticle tracking analysis, but also protein analysis. These basic technologies are all offered. And besides that, there are some more high-end and dedicated technologies like super resolution microscopy, for example, but also flow field flow fractionation analysis. So these are all technologies that we offer. And of course, there is also service, which we provide to academic users, but also to industrial partners. What I do see as a main advantage is that you can really advance the research area regional by gaining access to a much broader variety of technologies than as an individual host lab, but also connection to industrial partners. Like, they are really reaching out, whether it is to arrange demos for all the regional people working on extracellular vesicles and so on. So it's we are only officially recognized since April, so it's still very early. But the main concept is that we really provide access to different complementary techniques that allow both characterization, but of course also separation. So all different types of characterization methods, protocols that we have been working on, we have also available in the core facility. And finally, of course, we also offer support in EV track. That's also one of the aspects that we support. Yeah,
and we'll be talking about that very soon. So I think, an interesting segue from that is your work with bacterial EVs, because, they're quite a different, biology to the eukaryotic EVs that everyone's quite used to working with. Sort of how does that differ in terms of the methods you guys use?
Yes, so and this is actually a story I always like to share. We have been discussing the different methods that you can use to isolate extracellular vesicles, and I think there it's crucial that there is not one gold standard, because if everyone would use a one gold standard, it would definitely hamper novel discoveries. And we are very, how should I say, hardcore into depth in really trying to establish protocols that allow us to separate extracellular vesicles with high specificity or high level of purity from different types of biofluids. So how we came to the field of bacterial extracellular vesicles is that basically when we are localized in a university hospital. So most of the research that we do in our lab is focusing on patient materials. And in search of biomarkers, we often work with blood plasma samples. And from a biomarker perspective, when I discuss with clinicians, it is often the discussion, but a good biomarker doesn't have to be an EV-associated biomarker. And that is, of course, true from a clinical perspective. But from a research perspective, if you identify a biomarker you want to claim that it's an EV-associated biomarker, you need dedicated quality controls to actually confirm this. So we put a lot of effort in optimizing a protocol which combines a size-based separation with a density-based separation. And what you actually do with such a technological setup is that you fractionate your blood plasma sample in multiple size and density-based fractions. And this allows to increase the sensitivity towards biomarker discovery, especially if you do a downstream omics analysis, such as proteomics, for example. So for every patient sample that we were running in our biomarker pipeline, we not only focused on the fractions containing the human-derived extracellular vesicles that were of that size range and that density range, but we decided to really analyze all the different fractions. And what we could then observe is that in the human-derived extracellular vesicle fractions, we could definitely identify all the classical marker proteins that you would expect, such as tetraspanin, syntenin, alex, and so on. But there were higher density fractions where we were also identifying bacterial molecules, bacterial proteins, bacterial RNAs. And then we started to wonder, is this a contamination? Because many of the buffers, the kits that you use at some point have certain levels of bacterial contamination. So you have to start to really dig into detail. Are all our buffers free of bacterial contamination? Are they endotoxin free? And so on. So we first set up a whole pipeline where we validated the potential presence of bacterial contamination. If yes, we replaced the buffers. And by really scrutinizing the protocol and then combining different characterization techniques, including both omics, for example, proteomics, to demonstrate the presence of bacterial membrane proteins, but also known intraluminal proteins, with electron microscopy to visualize the presence of vesicular structures, with immunolabeling, for example, for OMP-A, to demonstrate that they are of bacterial origin. So this really took a long time, because it was the first time that it was actually being demonstrated that we can have bacterial EVs in the systemic circulation of patients. And then the reviewers ask for a lot of validations to be really sure that it's not a true contamination. So this was really a step-by-step approach. Now, what is striking is that we actually really don't know that much about bacterial extracellular vesicles in human body fluids or in environmental sources even. So most of the knowledge that is available in literature is really coming from in vitro cultured bacteria. So this is a whole new research area that is opening up, and that of course needs an additional level of attention, because it's not always straightforward. There are not many research tools available that can be used to study these bacterial EVs, so a lot of optimizations and developments are really required there.
Yeah, that really is quite a substantial finding. Like I remember when I saw that for the first time, bacterial EVs in blood. Blew my mind. And so are there, so far, has there really been any valuable, biomarkers or anything that has come out of these bacterial EVs thus far, these blood-bound ones?
Yeah, so, what we observe and reported, for example, is that it's patients that have an increased intestinal permeability, which can be cancer patients that are undergoing certain treatment schedules, such as chemotherapy or radiotherapy, but also other disease types, like patients suffering from HIV infection or inflammatory bowel disease. These diseases are associated with increased intestinal permeability, and in these conditions, we were able to identify an increased level of these bacterial EVs in the systemic circulation. Now, detecting them remains very challenging, because from what we at least quantify in our research group, they are like on average 1,000-fold lower abundant compared to the human-derived extracellular vesicles, which is already challenging to explore in a complex biofluid such as blood plasma. So there we do have some limitations, but it's definitely possible. The most important is that you really consider proper quality controls, that you ensure that you're really looking to a bacterial EV signal and not just, for example, LPS, lipopolysaccharide, which is present on the membrane of gram-negative bacterial EVs. Well, LPS is also present in a soluble form, being neutralized by lipoprotein particles. So just quantifying the amount of LPS that is present in a blood sample will not tell you anything about the presence or absence of bacterial EVs. So there are definitely some considerations that are needed when trying to evaluate them. Now, we know that they are released, but what we are currently really trying to explore is, first of all, which bacterial species are contributing to the release of these bacterial EVs. How do they reach the systemic circulation? And what is their fate and function like? Where do they go and what do they do? And how do they influence the host and disease processes?
Yeah, wow. That's working backwards from, just having the bacterial EVs in blood and not knowing what their source is. That is a thorny question. That's difficult. So maybe let's talk a little bit about the EV Track project now. Just for a start, for people who aren't familiar, do you want to give us a little introduction to what the project is?
Yes, of course. So EV Track, we actually initiated the concept in 2014 already. So then we made, in the first 4 or 5 years, we really focused on comparing different methods, optimizing protocols. And then we explored and observed, of course, that there is an impact of the choice of isolation method on the downstream analysis. And then we decided, okay, but maybe it's interesting now, what is the current status in the field? Like, how are these isolation or preparation methods being used in the field, what is the level of diversity and so on. So in 2014, and this was actually during an EMBO course, the first EMBO course on extracellular vesicles that I organized and ran in 2014, there we actually launched the initiative, Let's Explore Literature. So we selected 1,200 publications that reported isolation on extracellular vesicles. And we really screened these manuscripts for back then, it were 133 parameters that relate to the parameters related to the isolation methods that are being used to obtain extracellular vesicles from biofluids, and then also the different characterization methods that are being used, both at the protein level, at the particle level, and so on. And it took a very long time to read all these manuscripts because we read them one by one. It was, of course, with an international consortium. So I think we had approximately 60 to 70 people on the EV-Track consortium that really contributed to analyzing all these manuscripts for all these parameters. And then we processed these data. And what we could actually observe is that for those over 1,200 publications that we screened, that there are 1,000 different protocols being reported. And that, of course, was quite a striking observation, knowing that different protocols can end up and impact your downstream analysis. So then we thought, okay, we now did this literature analysis, but can we develop a valuable tool for the field that can help to raise awareness about the importance to report protocol parameters? Because otherwise it will be very difficult to reproduce experiments that have been made available in literature. So then we decided to build upon all the data we recovered from literature and develop the EV-Track as a transparency tool, so a tool that really supports transparent reporting, but on the other hand also centralizes knowledge on extracellular vesicles. So for the transparent reporting, the idea is that we basically ask researchers before they submit their manuscript to a journal for evaluation and potential publication, that they submit the experimental parameters, which they mostly summarize in the material and method section, that they upload these to the EV-Track knowledge base, and in return, they will receive an EV metric, which is a measure for the level of transparency in the material and method section. And based upon this EV metric, they get a very clear idea on which parameters have been well reported and which are currently missing. And by completing with the missing parameters, they can further increase the transparency and the chance for reproducibility for their work. So they can really use this EV metric as a coaching tool, first of all, to be aware which parameters really matter in my protocol and should I report so that others can reproduce our experiments. And then once the publication is accepted by a journal and becomes available on Web of Science or PubMed, the entry also becomes available on the EV-Track knowledge base, which means that we also collect information related to some EV biology, but also methodology, where we can easily follow up new trends that are occurring, for example, in types of methods that are being used, which can ultimately also guide us on how we should optimize, for example, ISEV guidelines, or are there new methods or biofluids for which ISEV guidelines may be or may receive an update. So that's the overall idea of the EV-Track knowledge base, but it's completely community-driven, which means that it really builds upon researchers that submit, and it's this information that gets curated and once published becomes publicly available on the knowledge base. Yeah.
So
And the tool was initially launched then in 2017. So that was the first effort. First, make sure that we get the tool and get it published and that the field is interested. But the second challenge is to make this tool then really valuable to the field so that people are using it, implementing, and that it generates valuable support and input for the researchers. And this is, of course, a challenge that we have been taking during the past 8 years, and which is why, we need some additional support and also looked towards developing a task force, because the difficulty with standardization and reproducibility efforts is really try to find the funding, because initially the tool, when it's not published and not available, it is highly novel and funding agencies will be willing to support it. But once it's out there and you're just looking towards improving, maintaining it, it's not interesting for them anymore. So finding the resources to really maintain it up to date and to ensure it's still a valuable tool for the community is really challenging.
yeah, that's the sad reality of this kind of thing, like meta research, which is sort of essentially what the EV-Track project is. It's valuable for the community and researchers at large, but it doesn't always necessarily produce, the new finding that the kind of thing that gets a grant or so it can be hard to get funding. I can see that, but it's really nice that this like two-way dialogue that sort of helps encourage a better quality of publication and a better experimental reporting. How many people sort of go that route? Because I suppose there's two different routes. There's people who do enter and communicate with you guys during the publication process, or rather right before, and then there's the reporting that happens on publications that are already out there. So how many people take that sort of first route?
So for the new submissions, so it's indeed important that you mention that it's communication, because what often happens is that you submit on so you generate a login, and then you're able to submit the parameters, and you then submit the document to the EV-Track administrators. But there is always a back and forth communication. Like often they will reach out to you if there are some uncertainties in responses that you submitted. But the other way around, if you have any questions and are not sure, you can send them and reach out to them via email, and they will also respond. So this is a very active back and forth communication. Now, one of the disadvantages is, I think for now we have close to 4,000 different publications that were submitted to the EV-Track knowledge base. If we look to the total number of EV publications that is available in PubMed, not including review manuscripts, of course, this is, of course, much higher. So we do capture, I think, by a maximum 5 to 10% of all publications that are currently available on PubMed. But at least we have to start somewhere. And so with the EV-Track task force, this is something we are currently actively trying to solve, is how to stimulate people really to use the EV-Track knowledge base more often. And the examples I always highlight is, first of all, when master students, PhD students, and even postdocs, when they start in the lab, they first and for all, like when you start to work on a project, you have to read literature. If they read literature, they have to submit the manuscripts to the EV-Track knowledge base so that they become familiar with the tool and that they also understand why it is important to use the tool. And after this testing period, let's call it like that, they always come back to me and say, wow, this was really valuable because now I read literature completely different. And indeed, how we read literature is we first check the material and methods to see how these experiments were were performed, is this really a valid approach, or should we be critical on the results that were obtained or reported in this manuscript? So the way that you process literature and interpret results completely changes when you start reading material and method section. When I started as a PhD, I skipped this. I never read the material and method section because you're interested in the results. And you read the abstract, you read the results. But knowing that the methodology that is being used can so profoundly impact the results and the conclusion you make, we turn it the other way around. And we first check what were the material and methods that were being used, and how can we interpret the results that were reported in the manuscript. So it's really generating an awareness about it does matter which material and methods were used, and try to critically reflect on it before you really start to think about results, because the one does not go without the other. So it can really help you to train new people that are coming to the lab. So that's one aspect. But then secondly, what we also do, and we actually did this for the bacterial extracellular vesicles, it's quite a new hot topic, emergent topic in the EV research field. And not many bacterial EV publications were available in the EV track knowledge base. So we decided to repeat our effort from 2014, but now with a really dedicated focus on this sub-area of bacterial EVs, where we selected over 800 publications reporting experiments on bacterial EVs, which we submitted to the EV track platform to really map what is the current status of this sub-area in the large EV research field. And so it can be very helpful as well to map the current status in the field, and then to identify what should we focus on, what is lacking, and how can we further ensure that the studies on bacterial EVs can really advance in the near future. So that is one other aspect. And then the third aspect is that you can also use it as a query tool. Like if you want to set up a protocol for a particular biofluid, and you're not quite sure, for example, how to set up your methods, how are you going to isolate the vesicles? Well, then, for example, you can go to the EV track knowledge base and explore. You can use different keywords or search terms. One of them can be a biofluid, and then you get a list of publications that reported on that biofluid, and you get an overview, for example, on which different methods have been used for their biofluid. And then you can start to do a comparative analysis, and it can help you also to guide and set up a protocol when you're starting to work in a different area. So there are multiple types of approaches for which you can use the knowledge base.
I think you're speaking to the right group of people, because, as you put it, that sort of two-way dialogue. If you're a student out there, and perhaps you don't have as many experienced EV researchers around you, as much guidance you can with your publication, reach out to EV Track and essentially get free support and recommendations on what you should do. So that is definitely something to encourage if you're a student listening to this. Do you have anything else to say maybe to any students who are looking at the EV Track, reporting, and maybe are worried about how to approach it? Are there any other resources that can help them along?
Related to EV Track, I basically want to mention, so it used to be managed completely by my lab, like a bioinformatician, a postdoc, PhD student working on that. But since 2023, so we moved towards an EV Track Taskforce, because the main aim, as I mentioned, it's difficult to get resources, so we have to try to come up with another structure that can ensure the maintenance of the knowledge tool. So we developed the EV Track Taskforce, where we have 3 working groups. So one working group is at the communication level. So when people submit data to the EV track. They do a first curation, communicate with the authors, and then when the publication is accepted and becomes publicly available, then they also do a final curation step. I think the EV Track Task Forces is a quite unique task force because we really also value the contribution of early career researchers. So Working Group 1 fully consists of early career researchers that received, of course, a training in it and now are actively involved in the process of really handling the submissions we receive at EV Track and also completing the curation. Then in the Working Group 2, so Working Group 1 is headed by Jan van Deun, who was the first author on the original publication and was really actively involved in developing the platform. And Working Group 2 is headed by Quentin Roux, and he also continued the effort on EV Track in my lab, is now back to a postdoc position as well. But although they are no longer in my lab, they are still very actively involved in the tool. And I very highly value and appreciate this because it also demonstrates that other people besides me think it's a tool that should definitely exist and be maintained. So in Working Group 2, Quentin and his team are really further developing the platform. How can we make it user-friendly and these aspects? And then in one of the aspects we are, of course, considering is how can AI help us to ease the process of submitting, but also of curating and maintaining the knowledge base. So this is something we are also actively trying to explore. And then in Working Group 3, we mainly have then more the senior level researchers that are trying to develop approaches on how we can stimulate the outreach, the visibility, the use of the platform. Different types of working groups. So if you have interest to contribute this, or if you have any suggestions after using the tool, please reach out and provide us with this feedback because we are very actively working to make this tool even better for the community. So every feedback you can deliver, we would highly value. And if you're interested to contribute, also please feel free to reach out.
It's funny you mentioned the number of EV track records and PubMed, because I sort of did a little, back of the notepad calculation in preparation for this. And yeah, the number I had was roughly 8%. And I looked back in time as well to 2023, and it was roughly 8% as well of EV track records to sort of the broader, number of EV related publications. So it's kind of kept pace with the massive growth in EV publications. So yeah, I guess you can't necessarily reveal things that are in the works, but what, you did mention AI there and using that to streamline. Is there anything we can maybe expect in the near future that, might put a bit of a dent?
We have actually the second version, update version of EV Track is ready. So we presented it actually during the ISEV 2025 meeting. There was also a booth where you could already explore the new website and the new tool. So there are definitely changes because the current EV metric, for example, was still building upon the ISEV 2018 guidelines, or at least aligning with the ISEV 2014-2018 guidelines. So the way that the EV metric is being calculated will even put more weight towards really transparent reporting the methods that you use. So less on which methods you use, but if you use the methods, that you really transparent report them. The the process of calculating the EV metric and the numbers of vectors that is accounted for will drastically change. And this is mainly because the first EV metric was very dedicated on did you perform this or that experiment or then the antibodies reported or not. So it was much more restricted. Now we try to implement or at least explore the full package of parameters. And I think this will also further help and stimulate the use of the EV metric. Maybe some aspect that could still hamper the use is that the EV metric may be looked at as a sort of a scoring of the quality of a manuscript, but I really want to stress and to highlight that this is not the case. It's really a tool that aims towards coaching. And sometimes, you will get an EV metric of 30% and not 80 or 90%, but that's also perfectly fine. Why? Because sometimes you're using sources for which you have limited availability and you're not able to do many types of experiments on them. Then, of course, the EV metric will be restricted, but that should not hamper you from submitting and using the platform, because it's absolutely not related to any type of quality or lack or whatever in the manuscript. It's really a matter of doing at least what is possible in your circumstances. And it will also help us to understand that and to even further improve future implementations of the EV metric and the EV track knowledge base. So this is really something I think it's very important to consider. And then the second aspect, if I can mention another one, is also the time. Like a feedback that we also received on the satellite event we organized at ISEV 2025. It takes a lot of time to complete the survey for EV track. Now, this is something we have been working on. And on the second version of EV track, it will go much more smoothly. It doesn't mean it will not take time to submit the survey. But, and I also discussed this here in my lab, you have been working on a project for 3 or 4 years. And if you then have to spend 4 hours or maybe up to 1 day to submit all this information to the EV track knowledge base, I really think it's worth the effort. If you can achieve more reproducibility and transparency with that, the 1 day would not add much of a time to the 4 years of hard work you have done as a PhD, a postdoc, or even a master student. Try to really consider this. It can massively help you. It can also generate additional visibility to the research you have done, because it also becomes available on the EV track knowledge base and pops up in queries that are being done. So please consider this. That would be my advice.
It's a good way to frame it. It's like spending all that time writing a paper and then, not doing a final spell check. I just want to pivot back to that bacterial EV publication and, maybe with a little bit of an aside too. So we did a little collaboration a while ago where we sort of made this map of the EV field and we sort of split it up into different areas and research focuses. And we combined that with EV track data. And, some of these different areas had very different coverage, like cancer research, for instance, had 26% and, other areas only had 9 and 10%, which I just thought, I thought was very interesting. And it was also a bit of a shame because, like you said, this is sort of gives us a meta perspective on what's going on in research in those areas. And when there isn't enough publications, you can't really make full use of that. But then, your bacterial EV publication took what that area only had 13% coverage to now it is, more or less fully covered, and you were able to say a lot of very useful things. So do you want to tell us a little bit about, that exactly the benefits of what happens when you do comprehensively have this kind of EV track reporting in an area?
Yeah, it gives a lot of information. You can really identify, because one of the striking observation is that 94 or 95% of the publications that we analyzed were really reporting on bacterial EVs from in vitro cultured bacteria, which again shows then, okay, but we don't know anything on bacterial EVs that are released in natural sources, whether it's human body fluids or other environmental sources. So that's a whole new era that is really ready to be explored. But on the other hand, if you then also look to the research methods that were being implemented, you could easily observe that it's very hard to combine different complementary characterization methods to characterize the BEV preparation you obtained from a particular source. Why? Because there are limited research tools available to really explore this. So, and what we actually did also in this analysis is we did a comparative analysis on what the impact of the publication of the ISEV guidelines is on all the publications that are available in EV-Track. And then since 2018, you can observe that there is an increase in the EV metric, meaning that with publication of ISEV guidelines, researchers become aware of the importance of particular methods and reporting them, and it also turns back in an increased EV metric. But if we do this analysis for the publications focusing on bacterial EVs, ISEV guidelines do not have any impact on the increase. So publications don't increase an EV metric after publication of the ISEV guidelines, which also shows us that the ISEV guidelines do not per se cover the needs of this sub-area focusing on bacterial EVs. And these are very nice aspects to consider, because is it because this area does not want to do the effort to comply with the ISEV guidelines? Not at all. What you can observe by doing this analysis is that the research tools are not there to be able to comply with the ISEV guidelines. So it really demonstrates that there is a need to develop research tools and to ensure that ISEV guidelines are also applicable to other areas, and that you can, at least by development of new methods, also comply with the ISEV guidelines. So this really helps you to understand what is the current status of a field, but also what are the needs of the field. So what do we need to be able to fully explore what is the composition, the biology, the function of those bacterial EVs? So therefore, we definitely need the development of new research tools. Another interesting finding also related to the nomenclature that is being used. So where it originally was mainly outer membrane vesicles, for example, we more often now observe the use of the overall term bacterial EVs. And at some point, you can also notice that even for gram-positive bacteria, some publications are reporting outer membrane vesicle as a term, which is actually a type of vesicle that is only being released by gram-negative bacteria. So there can be some, discrepancies and aspects also from the nomenclature point of view. So it's really good if you have a new area and you think it's worthwhile to understand what is the current status of this field. It's really valuable to use the EV-Track knowledge base to help and map this. It helps you, it provides the survey, first of all, so you can very easily complete all the parameters, but it can also help you to collect all the data and make some very nice graphs and conclusions out of that. We have had other researchers that actually reached out to us and that also on a particular field wanted to map the status, and then we can also help and assist on that. So if you have any idea on, I'm working on this particular area, but I feel it's not properly covered by EV-Track, please reach out to us so that we can discuss how we can help, how we can contribute, and assist you in making this happening. And of course, it will then also help to make EV-Track more broadly applicable over the different areas of EV research.
Yeah, it's funny you should say that. That's part of the reason I brought it up. This is the student network, and people are always looking for ideas and new angles on research. And yeah, that's a glaring one. There's plenty of areas of EV research that could do with a thorough EV track reporting. So if you want to sit in front of a computer and read papers for a while, get into it. Is there anything else you'd like to share about the EV track project and the team at the moment?
No. So as I mentioned, we are hard working towards increasing the visibility. So we are also thinking about, for example, establishing some webinars where we have online guidance on how the tool works, what you can use the tool for. So different training initiatives. We will also try to be involved in the more local national societies where we can organize short sessions to further help and assist you with that. But in the meantime, as I mentioned, I think it's really important if you have any ideas or feedback. First of all, if you didn't explore yet, please go to the EV-Track knowledge base and explore what it can mean for you and how it can help you. But also, if you have any feedback that you think we can take into account to further update and improve, please feel welcome to reach out. And it has been a huge effort to already keep the knowledge base up and running for the coming 8 years. I remember if we launched it with Jan, Jan told me, Anne, I'm not sure we can launch the tool, but what if people are not using it? So I told him during the past ISEV meeting, look, Jan, we did it. People are using it. It's even recommended in the MySEV guidelines. So I think it's really, really fantastic. And the effort after launching the tool is at least as huge as the effort towards launching the tool, because you cannot underestimate the time it really takes to maintain all of this. But I hope at least you find the tool valuable and supportive and can make good use of this. And this is for the EV track, but I also know from communication with Suresh Madhivanan, for example, the same accounts for Exocarta, Vesiclipedia, EVpedia, all these knowledge bases. In comparison, it's quite easy to establish them, but to maintain them and to keep ensuring that they are valuable for the community, that's the biggest challenge. But I believe with the EV-Track Taskforce, we have a very good team up and running now that is really further pushing the usefulness of the tool for the field. So this makes me really happy.
Wow, yeah, I feel like I could talk about this for hours now. There's so many interesting questions coming out of this, but we are starting to run out of time now, and I think that is a great note to end it on. I really hope that anyone listening to this who wasn't too familiar with EV Track is now a little bit more familiar and hopefully pretty inspired. I think it's a great initiative, and I hope everyone can see that and hopefully get involved a bit more. If you're a student who has a publication that you'll be hoping to get in soon. Thank you so much, Anne. It's been very, very interesting.
Thank you, Liam. I very much enjoyed, and I hope anyone listening to this podcast really has some key take-home messages to consider, which can further help you developing as a fantastic EV researcher in the upcoming years.